I'm David Britt in the Department of Biological Engineering. I'm an associate professor, and our research focuses on nanotechnology and the influence of nanotechnology in the environment. We use nanotechnology to build nanoparticles through reduction methods, so basic chemistry methods, to create particles that are on the size of a billionth of the width of a human hair. So with our research, we're interested in nanotechnology generally from two approaches. Some of our research focuses on using nanoparticles to direct protein shape. And so there's a number of diseases that are based on misfolded proteins in the body, such as Alzheimer's and Parkinson's, and in agricultural mad cow disease. So from the application standpoint, we look at how do nanoparticles that we've synthesized in our laboratory and characterized with advanced techniques in our laboratory, we want to know how those can be applied in medical settings and agricultural settings. We also focus on research that wants to understand what is the long-term impact of nanotechnology in environmental settings. Nanoparticles are found in many common consumer products, from paints to shampoos to cosmetics. So we see these are being introduced into a lot of products without knowing fully where these products will end up. And so we use advanced imaging methods to look at nanoparticles and characterize their size, and more importantly, how that size might change in biological settings such as the environment. And so we're talking about very, very small particles that can't be seen with traditional imaging techniques such as optical microscopy. So we use atomic force microscopy in our laboratory. The atomic force microscope, we have a two-microscope system. We have an optical microscope, which many people are used to seeing, and it's a concept that is easily understood, where we use transparent lenses to focus visible light onto our sample. However, that type of microscopy is limited by the wavelength of light. So sitting on top of our optical microscope is what's known as an atomic force microscope. And this is capable of imaging down to the nanometer scale. Our research is very interdisciplinary. We have a team of engineers, microbiologists, geochemists, and plant biologists. So this is very diverse research, and our students that work on these projects are exposed to a wide variety of techniques and topics.
My name is Rachel Johnson, and I'm a biological engineering major.
I'm Joshua Israelson. I'm a freshman in the biological engineering program.
I'm Kyle Isaacson. I'm an undergraduate working in Dr. Britt's laboratory.
My name is Alexis Morris. I am a sophomore in the biological engineering program.
In high school, I did an internship in an industry lab, and I got really involved in that, and I liked it a lot. And so when I got to college, I applied for a dishwashing job in a lab, and then it gradually just progressed into a research job.
Well, I study the effects of zinc oxide nanoparticles on the formation of protein aggregates, particularly the types that produce proteinaceous diseases like mad cow.
And my research was seeing how nanoparticles can be, if they can be capped with phosphate and ozone.
I am going to a conference soon. I'm going to Nano Utah, which is on October 11th and 12th.
My poster is called... What is it called? Characterization of zinc oxide nanoparticles.
The name of my presentation is PEG-silane modification of zinc oxide nanoparticles for neurodegenerative disorder treatment.
Biological engineering is inherently an interdisciplinary field, so our students enter this program usually with the desire to build on engineering skills, but have a broad knowledge in the life sciences such as biology and chemistry.